Okay, and welcome back to part 3. We're talking about general cell respiration. Next question. Major nutrients are degraded to what? Acetyl-CoA, but some are reversible pathways. Name 4 nutrients that are converted to acetyl-CoA. Carbohydrates, fats, proteins, and alcohol. What happens to the carbons of acetyl-CoA in the mitochondria? What is then transferred to the coenzymes? They are oxidized to CO2 via the TCA. What is then transferred to the coenzymes? Hydrogen. Electrons are transferred to the respiratory chain. Under low energy conditions, how is fat utilized in the brain? Via ketone bodies. They can generate acetyl-CoA. Acetyl-CoA can also act reversely to make ketone bodies, fatty acids, leading to triglycerides and amino acids. How much of the cell's energy comes from the mitochondria? About 95%. How many ATP does glycolysis yield? 2 ATP, and it's anaerobic. For every mole of glucose, how much ATP is generated from the mitochondria? About 36 ATP, and that's aerobic. What happens after pyruvate and fatty acids and amino acids are transported into the matrix? They are oxidized to generate CO2 and the reduced nucleotide coenzymes NADH and FADH2. What is unusual about the mitochondria compared to the ER and Golgi? It has separate DNA, makes its own ribosomes, and has a double membrane. What is the only other organ in the body with a double membrane? Nucleus. Where is pyruvic acid converted to acetyl-CoA? In the matrix of the mitochondria. Fatty acids and amino acids are converted to what in the mitochondria? Acetoacetic acid. How many molecules of CO2 are produced for each acetyl residue? 2. What is the final common pathway for the oxidation of all major nutrients? TCA. Where does the final oxidation take place? In the mitochondrial matrix. What does the first reaction of the TCA form? Citrate, which is 6 carbons. Describe the formation of citrate in the TCA. The acetyl group of the acetyl-CoA reacts with the 4-carbon compound oxaloacetate to form the 6-carbon citrate. The acyl group reacts with 4-carbon oxaloacetate to form 6-carbon citrate. What enzyme catalyzes the irreversible, cannot go in the opposite direction, reaction of the oxaloacetate to citrate? What is the enzyme there? One of the most important, of the most top 3. That's citrate synthase. What is then done with the citrate? The remaining reactions regenerate oxaloacetate from citrate, with 2 carbons released as CO2. The term amphibolic is used to describe a biochemical pathway that involves both catabolism and anabolism. Why is the TCA described as being amphibolic? Because of its catabolic and anabolic nature, energy production and biosynthesis, not just making electrons, but also synthesizing other products. NADH produces 3 ATP during the ETC with oxidative phosphorylation because NADH gives up its electron to complex 1, which is at a higher energy level than the other complexes. When complex 1 transfers the electron to complex 3, energy is given off to pump protons across the membrane, creating a gradient. The electron moves again to complex 4. So it's 1, 3, and 4. And again pumps more electrons across the membrane. Because NADH started with complex 1, it had more chances to pump more protons across the gradient, which powers the ATP synthase and gives us 3 ATP per molecule of NADH. FADH2 produces 2 ATP during the ETC because it gives up its electron to complex 2, bypassing complex 1. By bypassing complex 1, we missed a chance to pump protons across the membrane. Remember, protons are the hydrogen ions. So less protons have been pumped by the time we get to complex 4. The protons still have to be pumped, enough to fuel 2 ATP created by ATP synthase. Okay, we'll get into a little bit more detail now about the citric acid cycle. What is the intermediate for carbohydrates in the TCA? Malate. What is the intermediate for proteins in the TCA? Aspartate or glutamate. What is the intermediate for fatty acids in the TCA? Citrate. Why are redox reactions, which are electron gain, involving coenzymes important in cellular respiration? FADH2 and NADH donate hydrogen ions, protons, and electrons to the electron transport chain in the inner membrane of the mitochondria. As the electrons pass from one carrier to another, which is a redox reduction, which equals electron gain at oxidation, and then equals electron loss, hydrogen ions are pumped into the space between the inner and outer membranes, creating a hydrogen gradient with the stroma of the mitochondria. The hydrogen ions pass through the membrane-bound ATP synthase to the matrix of the ATP, and ATP is produced. So if there were no redox reactions in the ETC, there would be no large-scale generation of ATP to the whole point, which is the whole point of respiration. The byproducts of most catabolic processes are NADH and FADH2, which are the reduced forms. Metabolic processes such as NADH and FADH2 to transport electrons in the form of hydrogen ions. Okay, next we're going to talk about redox coenzymes.